Welcome to those of you who have joined today's webinar. I'm Maria Williams with the Collaborative on Health and the Environment. CHE enjoys bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's partnership webinar, which is titled Down the Drain with PFAS, the latest on testing, measuring, and mitigating community water contamination. Our moderator today is Dr. Wendy Heiger-Bernays, Research Translation Core Leader of the Boston University Superfund Research Program. We will leave time following the presentations for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentations. After the presentations, our moderator will read out questions for our presenters to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 60 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Wendy.
Thank you, Maria. Welcome. This is the 19th call in a series that is co-organized and hosted by Collaborative for Health and the Environment and the Boston University Superfund Research Program. The National Institute of Environmental Health Science funds Superfund research programs, which are multidisciplinary programs with centers around the country. Each of these centers conducts and communicates research with the goal of breaking the link between environmental exposure and disease. This research program is truly unique in that it has always emphasized solution-oriented, multidisciplinary approaches, research translation, and community engagement. CHE is a partner on the BUSRP's Research Translation Core, and on each call, we highlight work that is being done that is relevant to the centers across the country for CHE's membership and listeners. I do want to recognize the newly funded URI Harvard SRP that is focused on PFAS, and we look forward to learning and working with them as they answer some of the questions that will likely be asked today. After my brief overview, I will introduce each speaker who's asked to speak for about 13 minutes, and their complete biographies are located on the CHE Call website if you want to know more about them. As Maria mentioned, we will send you a very short evaluation survey after the call, which will help us tailor future calls to your interests. And I request that you take the few minutes to respond. So today we're bringing you part 2 of a call that we organized a little over a year ago on perfluoroalkyl substances, or PFAS, with a focus on community response to drinking water contamination. These are a large group of largely unregulated manufactured compounds that are used to make consumer and industrial products stain or grease resistant, water stain or grease resistant. At the consumer level, PFAS are used in clothing, cosmetics, carpets, cookware, and food packaging. They were, and in some cases are still used in firefighting foams at airfields. Today, we will bring you up to date on this topic that is receiving a great deal of attention by state and federal regulators, researchers, healthcare professionals, and community members. Most of the work to date has focused on water supplies, but the full extent of PFAS contamination and human exposure remains uncertain. PFOA and PFAS, the older and larger PFAS, have received the most attention and will not banned or phased out of production in the US, in places, and in Europe, but are still in production in China. However, they and other PFAS remain present in our indoor environments, food, and in some locations in water supplies. They are measured in our blood at levels that appear to be decreasing. This is PFOA and PFAS, but the replacements may be increasing and remain insufficiently tested for toxicity and are unregulated in commerce. Efforts are underway in multiple states to restrict or ban some of the PFAS. In 2016, the US EPA established drinking water health advisories for PFOA and PFAS. While these are not enforceable standards, they provide a measure against which concentrations of these chemicals in drinking water can be compared. Since that time, 14 states have adopted the EPA guidelines, and 4 states have expanded regulation to include multiple other PFAS that were measured in public drinking water supplies across the country. People on private water face additional challenges due to the fact that these systems are unregulated by the federal Safe Drinking Water Act. On our first call, we focused on the community organization around PFAS, contaminated water supply in Vermont, the challenges of testing for PFAS in water, and what was known about the health effects. The focus of this call is to provide information for communities and others facing PFAS contaminated water issues, being as practical as possible, recognizing that this field is rapidly evolving as more of the science becomes available. The speakers on this call are Dr. Nancy Rothman of New England New Environmental Horizons, Mr. Richard Spiese of the Vermont Department of Environmental Conservation, and Dr. Thomas Webster of the Boston University School of Public Health. We hear first from Dr. Nancy Rothman, who is CEO and Principal Scientist at New Environmental Horizons. And in the interest of time, I point you to her bio on the website. Dr. Rothman will discuss measurement of PFAS in water and other environmental samples and will identify challenges to interpretation of contamination test results. Turn it over to you, Nancy.
Thank you so much, Wendy. Can everybody hear me? Yes? Okay. I'll be talking about the measurement of PFAS. And the first thing, Wendy mentioned is that PFAS has been produced for a long time and has a wide range of applications. And this has meant that PFAS has gotten into our environment, into the water, into the soil and the sediment, into the air. And now we're looking at how we're going to be able to evaluate the extent of the PFAS. This slide from our friends at SGS Access is not meant to blow your mind, but it shows you some of the chemical structures of the compounds that are of concern. Up in the upper left-hand side, these are the perfluorocarboxylic acids. Next to them, the perfluorosulfonic acids. And down below, starting with FTS, are precursors. And these precursors are important because they can break down to form the carboxylic acids and the sulfonates. Now, I wanted to just point out that what I'm showing you is really the straight chain structures, meaning that if you look at the FTOH at the bottom, the carbons are linked together in a straight line, like a backbone. However, the carbons can actually link in different ways called branching. And so that, for example, for PFOA is not a single compound, which you might have expected. Instead, there's actually 11 compounds that represent PFOA, 1 straight chain and 10 branched isomers. So you can see if we account for all of the straight chains as well as all of these branched isomers, we have many, many compounds to consider. Method 537 is a drinking water method only and is really the only method on the books that is routinely used for the analysis of PFAS as an EPA method. There's no recovery correction, which I'm going to discuss a little bit later, as well as there's limited number of compounds that are looked at by method 537. The ASTM methods, there are 2 of them that came out in 2017. They're not widely used, but they look at matrices other than drinking water, but they don't allow recovery correction, and but the analyte list is expanded. These other methods, which I don't have time to discuss in full, TOP, PIGGY, AOF, CIS, can be used in combination with other analytical methods to determine the overall extent of the fluorine contribution to a sample. For example, the TOP looks at the precursor potential, meaning those compounds that I showed you at the bottom of that slide a couple of slides ago, if they have the potential to oxidize to become the carboxylic acids and the sulfonates. So these methods can help, along with the other methods, to really look at a sample in detail. Now, laboratories have modified methods which for matrices other than drinking water, and I want to point out that these modified methods are not the same from one lab to another, so it's important to understand modifications depending on the matrix. The ITRC, the Interstate Technology and Regulatory Council, of which I'm a member, has created fact sheets for PFAS. The top first 6 fact sheets from history and use through the remediation technologies are available, and the website is available on the URL is available on the website for this talk. And these fact sheets try to impart some information about what's been happening to date with PFAS analysis and regulations, etc. The last fact sheet, the AFFF foam, which as Wendy's explained is a firefighting foam, will not be available until the summer. But these fact sheets form the basis of an introduction to PFAS for users, and are being expanded to create a technical and regulatory document that will be available in 2019. I was in California a couple of weeks ago working on the site characterization, sampling techniques, and lab analytical methods part of the document. The Department of Defense and Department of Energy have a quality systems manual that's been issued, 5.1, and within that manual, there's a table B15, which focuses on PFAS analysis in matrices other than water, and it establishes the quality control that is required for laboratories to adopt to produce data that is compliant with DoD and DOE. This table is really very interesting and can help guide you to understand the methods of analysis and whether or not an analysis is of acceptable accuracy and precision, etc. And again, the URL for this manual has been given on the website for the CHI website. Now, what type of data do you need? I've heard that some people just get a results sheet for their samples if they're doing drinking water, and I would encourage you to not accept that as being acceptable. Instead, I would ask you to look for a summary data package that has a cover letter or a lab narrative that explains the analyses that were performed and whether there were any deviations or problems that occurred. Then the results for your samples, and then results for all quality control. The laboratory methods, for example, method 537 requires that the laboratory perform method blanks and laboratory control samples. And these are QC samples to monitor accuracy for the protocol. And you should ask the laboratory to produce these and report these data to you so that you can look at your overall data and see if there may have been an issue. For sites that have critical samples or that are looking at a large-scale evaluation of PFAS, full deliverables may be required, which would be an extra expense for you. But these contain all of the raw data that's associated with the analysis and can be used to look at an in-depth review of the quality. The factors affecting analytical results are background contamination. Many PFAS results have been adversely affected by the fact that the field collection activities have not understood that PFAS was ubiquitous. And so early on, before people understood this, there would be contamination of samples just based on the environment that the samples were being collected in or the materials that were being used. So background contamination in the lab as well can happen, and so that it's very important for this to be minimized. Quantitation of the branched isomers along with the straight chain. Well, in 2016, the EPA issued an advisory indicating that the branched and the straight must also be quantitated or reported along with the straight chain isomers. But prior to that, some laboratories only reported the straight chain or the linear isomer. So as I said, if you have 11 isomers for PFOA and only one of them is a straight chain, if data produced prior to 2016 only reported the linear form, your data may have been biased low. There are also interferences that can be caused by the matrix. This is not something that you would know, but if you were sending an unusual matrix, for example, an egg sample or a particular soil, non-drinking water matrix to a laboratory, it's possible that there are interferences that can cause enhancement and suppression of analytical signals. And that's something that the laboratory has to be aware of and is monitoring as they're running unusual samples. Not using recovery correction can affect accuracy. Method 537, the ASTM methods, do not permit isotope dilution, but the DOD and DOE, that table I told you about in table B15, they require isotope dilution. So we're at a little crossroads here on the methods. Reporting issues can be a problem. If you have a compound name that ends in ATE, that's the anion form of the compound. If it ends in acid, that's the acid form. If you're comparing data between laboratories, or you're comparing your data to a regulatory guidance, you want to make sure that you're comparing the compounds on a apple-to-apple basis. Usually the acid form is in the regulation, so that you've got to make sure that you're looking at the right compounds and the right data. Temporal issues can affect sampling analysis to a great extent. If samples are collected in the winter versus the summer, and there are problems that can occur in the laboratory if the extracts are not analyzed quickly enough, etc. So all of these factors, or in combination or singly, can cause problems with your analytical results. Wanted to touch on this because this is an important issue that I think is underestimated in some data. Prior to the extraction or preparation or analysis of samples, surrogate compounds or isotopes are added to monitor whether or not whether the accuracy of the preparation analysis were acceptable. For method 537 in the ASTM methods, surrogates, the top bullet, are added to samples, and the recovery for those surrogates infer the accuracy for the preparation and analysis for all the compounds that are reported. In isotope dilution technique, a labeled isotope of a target compound, so for example, a C13 PFOA isotope, is added to the sample prior to preparation analysis, and the loss of this isotope will mimic the loss that potentially would happen for the compounds of concern. When we use these isotopes to quantitate, which means to calculate the concentration of the unlabeled compound, we essentially recovery corrected the data back to 100% accuracy. So the isotope dilution technique is a much more accurate way of evaluating data. Analytical recommendations. You want to make sure that the lab has performed the method of extraction analysis on the type of matrices that you're sending to the lab. For non-drinking water samples, for samples that are analyzed not by method 537, if at all possible, isotope dilution and recovery correction should be used. It may not be used, once again, for drinking water analysis. A laboratory may not modify method 537 at all if they're doing drinking water. It must be done exactly as written. If possible, and you're doing, extend the list of analytes, the C4 and C5 alkyl acids can tell whether or not there is a plume that's developing for, the C8s or C6s are coming along. And precursors, if the analyte list can include those, it can tell you whether or not you have potential for the carboxylic acids and sulfonates to be formed. The laboratory must modify their techniques to eliminate all PFAS background, and must always report the branched isomers. If you wanted to, you make sure of this when you talk to a laboratory. You say, I assume that you're reporting both the linear and the branched isomers, and then they'll know that you are a little bit informed and make sure that that's being done. And if you can, you want to perform quality control above and beyond what the methods require. Field blanks can help to understand whether there was contamination that was ubiquitous in the field that's not connected to your samples. Field duplicates can help understand whether or not your results are meaningful. So conclusion,
Yep.
The field of expertise is expanding very quickly, and quicker than I'm actually going through these slides. There's a lot of resources out there. As I said, the ITRC fact sheets, the QSM 5.1. There are updates in next year. The EPA will have isotope dilution methods available. Use labs with experience, minimize contamination, and make sure that you look at the data to evaluate the quality of your results. Thank you very much.
Thank you very much, Nancy. Let's now hear from Mr. Richard Spiese, who's been with the Vermont Department of Environmental Conservation Waste Management Division since 1987. And I think the past year or 2 years, he has devoted his time to investigation into PFAS contamination in Bennington, Vermont, which continues today. And he'll share his expertise on water treatment systems for removing PFAS in this community. Richard?
Yes, good afternoon, everyone. This is Richard Spiese with the Vermont Department of Environmental Conservation. And as said, I've been working on Bennington since February of 2016. And since then, we've done quite a bit of sampling and maintenance of point of entry treatment systems. And I'm going to share some of the information that we've gleaned from that work in this presentation. So this is how it all came about. Most of you who have been to Denver recognize this as Denver International Airport. And the material on top of the airport that forms that tent is what was produced in Bennington, Vermont. It was produced for many applications, including the Superdome. And the largest project that I was interested in anyway was a coating that was produced for the Saudi Arabia to go to Mecca and cover the pilgrims coming to Mecca to see the Quran. And they produced 100 acres of this material at that facility. This is an aerial photo of Bennington, Vermont. To the west, or the left side of this map, is the state of New York. We didn't go over there and sample. Our counterparts in New York had done that. And to the right is the Green Mountains, and it's a state forest or national forest there, and so there are no water supplies up there, although we have sampled some springs located at the top of those ridges. And to the north is another town where we've only found very limited contamination. And to the south is either public water systems or, again, we did not find contamination. And the way you can tell that is the blue dots are non-detects in our water samples, and the other colors are just more and more higher and higher concentrations of contamination, with, of course, the beautiful red being the highest level of contamination in water supplies. So in our effort down in Bennington, we've sampled almost 642 water supplies. About 60% of those had some level of PFOA in them. And I do have to make sure I clarify that the main contaminant of concern that we were interested in is PFOA. There was some PFHPA identified in our sampling, about 10% the level of PFOA. That's the 7-carbon form of PFAS. About 235 samples, 37%, we did not detect. Of the samples that we took, we did not detect any contaminants. And as I said, 318 were greater than the Vermont regulatory standard of 20 parts per trillion, PFOA, PFOS combined. And those are the ones that I'll be speaking about where we put point of entry treatment systems on them. So one of the immediate things we did the very first day we identified the problem was we had to get bottled water to the people there so they would stop drinking contaminated water. And obviously that's the most important thing that you can do, is to make sure that people stop drinking their water if the water is found to be contaminated. So as I said, we immediately provided bottled water. Over the course of the next year or so, 13 homes and businesses were connected to the municipal water system where there was already a water supply line going by the homes. And the rest of the homes, as of the middle of last month, we put treatment on the wells. I think it was 318 point of entry treatment systems, although a couple of the homes did get point of use systems for specific reasons. And I can't remember if that's inclusive or exclusive. And we felt that the long-term solution was to try and get municipal water to all of the homes. But until that could be done, that we'd have point of entry treatment systems on them. There are some homes that will not be connected to municipal water, and they will remain on point of entry treatment if we cannot find them clean water in some other way. So these are what these systems look like. On the right-hand side of the picture, you can see the blue pressure tank. For those of you on municipal water, you may not have seen those before. The water goes in there and gets pressurized and pushed to the home. As the water comes out of there, it goes through a small filter, usually 25 micron filter, to remove any particulates that could clog up the carbon. That's the little blue cylinder just to the right of the sign that says Culligan. And then the water goes out through there and then through the 2 granular activated carbon canisters. The first canister is called the lead canister, and that it gets the contaminated water first for treatment. The second one is the lag canister. And then after that, it goes out of that and through another, usually 5 micron filter to remove any sloughing that might occur. And we haven't really seen much of this, but certainly read about the possibility that if the water sits for a long time in contact with the carbon, you get a little biofilm that gets produced, and it can then come off all at one time. That's the slough. And so we want, it would not be, it's not harmful in any way, but it would not really be something pleasing for people to drink. And so we have another filter to remove that. And then right just to the left of the window that you see in the picture is a totalizer that will monitor how much water goes through the system. And then the water actually goes up and around the window, and just above the container that says Culligan on it is a stainless steel cylinder, about 2.5 feet long. That's a UV lamp, and that's installed to destroy any biological contaminants that could be produced in this process to make sure that the water is completely safe for drinking. And then it goes into the house for consumption. And I included a schematic with some of the specifications on it, really for when it gets put up on the website. So right after the system is installed, we set up with Saint-Gobain, the responsible party for much of this contamination, a sampling schedule. And within about a week or so of the system being installed, the consultant for them would come in and take their first round of water sampling, and they would sample the influent water and the effluent water. And one of the things we were potentially concerned about was some reporting that had come to us from New York State that there was some arsenic that could be created as part of this, and I'll get into that in a few minutes. And so we would take an arsenic sample to make sure, because that's another obvious contaminant of concern. And then a month later, there would be a second round of sampling of the influent and midpoint. If originally there was no contamination coming out of the effluent of the system, the contractor for Saint-Gobain believed that sampling from the midpoint would be sufficient. We negotiated with them and said that would be fine, that they didn't have to sample all 3 ports, the influent, the midpoint, and the effluent, but that if there was breakthrough of any of the carbon canisters at the midpoint, they would have to immediately go and take a sample of the effluent so that we could report to the homeowner what was actually getting into the home. And then if the first round of water sampling failed the standard for arsenic at 10 parts per billion, that they would have to then go and take a second arsenic sample at that time to see if levels would come down. And then the third round, they would do the same thing, influent and midpoint, and if we did indeed have any arsenic exceedances, they would have to sample for that again. So I included this slide from Calgon Carbon, who's I think the largest manufacturer of carbon in the world, from their webpage, to show that the green graph on the top is PFOA coming into the system, and that is in micrograms per liter. And actually, since it's 0.35 to 0.4 micrograms per liter, you could also say that's 350 to 400 nanograms per liter, or parts per trillion. And then the dark blue graph from the bottom of the screen going up is a total organic carbon, and that scale is on the right-hand side, and that is in milligrams per liter. So it's to show that there are a lot of other things being removed by the carbon other than just PFOA, and that after operating this system that they operated for up to 600 days, they did not see any breakthrough of the contaminants of concern in their carbon I obviously have some data that's a little different, but I thought it was important to show that as a baseline. So one of the other things was, how are we going to maintain these systems? And Culligan Water Treatment Company, who was hired again by the responsible party, set up a schedule to get to all of the residences in Bennington, and they will be replacing those paper filters to the left and right of the carbon filters every 4 months. They replace the ultraviolet lamps every 12 months to make sure they have enough energy to actually kill the bacteria. And depending on the hardness of the water, they would then be cleaning the quartz sleeves that the ultraviolet lamps sit in. And they do that from 1 to 3 times a year. And then regardless of whether there's any breakthrough in the carbon or not, they felt it was important to, and we agreed to replace the carbon every 2 years to make sure that the carbon was fresh and operating appropriately. So in the long-term monitoring of these systems, which we have been doing, that we base that on the influent concentration coming into the system. And for the higher levels of contamination at the site, over a part per billion, they agreed to sample every 3 months. And for the middle levels of contamination, between 200 parts per trillion, or 10 times our standard, and a part per billion, they would sample twice a year. And for influent concentrations that were less than 200 parts per trillion, they would sample once a year. We did negotiate that there could be a change in that frequency, and based on some of the work we've done, we think that maybe it's just as important to look at water usage as it is water contaminant levels. So there's a change out process. I'll go through this very quickly. You know, they take out the lead canister, they move the lag canister up, they replace the lead canister. A lot of questions come up about what do you do with this carbon. They compile all the carbon into one large 1-ton group and send that back to Calgon to make sure that it's properly incinerated. And then before they do that, they look at the canisters to see if there's any what's called channeling or biofilming that could explain why systems aren't operating as efficiently as they should be. Some of the initial influent results that we got very early in the process, the highest level of contamination that we saw in any wells was 4.6 parts per billion. And coming out of the system, we did not see any PFOA originally. As I mentioned, we did see some arsenic in some homes, about 10%, up to 32 micrograms per liter, which is much higher than our drinking water standard. As fresh water flushed through these systems, the arsenic level dropped down in almost every case to less than 1 microgram per liter. Although I don't know exactly why that occurred, my working hypothesis is that in the creation of the carbon, it's created in a reduced environment, and so the carbon is reduced when it's shipped. And because it's reduced, the arsenic is mobile and will then move through the system. As you flush fresh water through the system, that water is oxygenated and will slowly turn the conditions in the carbon canisters from a reduced environment to an oxidized environment, which makes the arsenic immobile, and it doesn't move through the system anymore or into the water and into the home. So currently we've taken over 1,600 samples from homes over well over 2 years in time. Of those, we've had 14 samples show some level of breakthrough where we've seen PFOA coming through that first carbon canister. The average concentration was about 7 parts per trillion, with a range from 2 parts per trillion to 11 parts per trillion. And the average amount of water coming through is about 100,000 gallons of water when we saw that breakthrough, with a range of 24,000 gallons to 224,000 gallons. And based on those results, again, at a cursory level, I think that the amount of water coming through is just as important as the concentration. Some of those concentrations are relatively low where we've seen breakthrough. As I mentioned, the arsenic levels, we've taken over 244 total samples originally, saw very few breakthroughs, about 31, or a little over 10 or 15%. The second time we sampled a month later, only 3 locations showed very little arsenic coming through, but just above the standard. Over time, over the next month samples, those all went to less than 1 microgram per liter. So to correct that problem and make sure as we changed out carbon, we didn't have that as a problem moving forward, Culligan is pre-washing all of their, although the carbon is pre-washed by the manufacturer, that still doesn't seem to solve that problem. Culligan's just flushing fresh water through it, several hundred gallons, and then taking an arsenic sample before they actually bring it out to the homes and install it. And since then, we haven't seen that problem reoccurring. So that's all I have, and I'll turn it back over to our next speaker.
Right, thank you very much, Richard. That was extremely informative. So what do we know about the health effects of PFAS on people? To address this, we'll hear from Dr. Thomas Webster, who's Professor of Environmental Health at the BU School of Public Health, and who's an environmental epidemiologist and exposure scientist, whose main research areas includes understanding exposures and health hazards of chemicals in consumer products and mixtures, including PFAS and other chemicals. I'm just going to turn it over to you, Tom, because we're running a little short on time and we want time for questions. So,
all right,
there you go.
See the screen?
Yes.
Okay, excellent. All right, so just a quick reminder of things to know about PFAS for the audience. This is a large and complicated class of compounds, and it's a moving target. So what we're seeing now is different from what we used to see. They occur as mixtures, and they're structurally related to fatty acids, which is one of the reasons researchers are interested in metabolic effects, things like obesity and diabetes and cholesterol and that sort of thing. In terms of the human serum levels, they are widely found. North Americans are approximately the same as Europe. Men tend to have higher levels than women. And the graphs that you see come from the CDC. It's a representative sample of the US population, and these are the four of the PFASs that are currently measured in serum. And you can see most of them are going down. There was sort of an uptick in the PFNA, the green one, but that seems to be going down. These are only the ones that we're measuring. Things in whole blood can differ from serum, and we know there are unidentified organofluorine compounds in human blood, as I think Nancy mentioned earlier. There's some ways to look at that. In terms of exposure, for epidemiology, we typically measure exposure using samples of serum or plasma. And the levels that we see in people's blood reflect exposure to the compounds we look for, as well as precursors. Again, Nancy brought that up. For most people, exposure is estimated to be predominantly diet, followed by indoor exposure and water, small. Although for some populations, water is clearly a dominant source of exposure. Although I do think these estimates need to be updated because there's been a lot of water information recently. All right. In terms of health effects, I'm not really going to talk about toxicology today. I'm going to talk about the epidemiologies, which is when we study effects in people. And there's really been an explosion of research published since we last gave this call, just a year and a half ago. So there's been about 40 published PFAS epidemiology papers since then. And again, metabolic effects, things like obesity, lipids, diabetes, birth outcomes, neurodevelopmental, some immune system stuff, and then some other things, thyroid hormones, reproductive effects, etc. And again, this is, it's too much for me to talk about all these. And I'm going to briefly discuss some of the new studies, particularly ones coming out of Project Viva. And I'm not trying to provide a full literature review evaluating all this new studies. We're sort of still in the trenches in the epidemiology. All right, so a few things to know about environmental epidemiology for those of you who aren't familiar. First of all, epidemiology, study of effects in humans, and that and animal toxicology are complementary. They tell us different facets of the same story. Epidemiology is hard and expensive. And if you read epidemiology papers, a good paper should discuss the strengths and weaknesses of the study. And that's because none of these studies can be done perfectly. They always have good parts and bad parts. The proper design of studies and the methods to mitigate weaknesses is a major part of what we do. And probably the most important thing I can tell you is that because of all these issues, environmental epidemiologists rarely, if ever, rely on a single study or paper. Evidence can be inconsistent between different groups, and it typically takes a substantial body of studies to come to scientific conclusions. Now, that doesn't mean we can't do policy in the meantime. It just means the epi sort of takes a while, and it takes a while to sort out what it all means. A few other things that you need to know to understand what I'm talking about today. A cross-sectional study is where you measure exposure and outcome at the same point in time in a person. So for example, you measure their blood level and you determine whether they have diabetes. A follow-up study is we measure exposure at the beginning, and then we follow people over time to determine who develops the outcomes. So the second kind of study is much stronger. Again, just sort of really basic epidemiology so you understand what I'm talking about. What we really like to determine is whether PFAS exposure is causing a health outcome. But a few of the things we need to worry about in the real life, amongst others, is it possible for the health outcome to actually affect the levels of PFAS that we see in blood? This is called reverse causation, and it can occur in cross-sectional studies. And because of the sort of peculiar properties of PFASs, we do worry about this. Another thing you might have heard about is confounding. That's when another factor could be changing the PFAS exposure level, say that we measure in blood, and the health outcome. And these can be things like behavior or physiology or socioeconomic status and all sorts of things. So epidemiologists worry about all these things in great detail. Let me say a little about what we did in Project Viva. This is a prospective cohort where we recruited about 1,000 mother-child pairs in the Boston area. The exact numbers depend on the particular study. We recruited the moms back in 99-2000, which was the peak serum levels for things like PFAS and PFOA. We measured PFAS in the mother's plasma early in pregnancy and again in children when they were about 8 years old. The primary focus will be on the major PFASs, PFAS, PFOA, PFNA, and PFHXS, the hexasulfonate. And we followed the kids over time and we examined birth outcomes and then child health. This was a large, very well-designed study. In terms of some of the results that we found, we looked at preterm birth and birth weight. These are both important for child health. And we found that prenatal PFASs were associated with an increased risk of preterm birth. They were also associated with decreased birth weight. And this was not confounded by maternal physiology. So there's been quite a big debate in the literature about the effects of kidney function on both the levels of PFAS we find in the mom's serum and its connection to birth weight. So that could be one of these confounding factors. We've looked at thyroid hormone. Oh, and by the way, the names that I list, those are the names of the articles, and I have the references at the end of the presentation. We looked at thyroid hormone disruption. So as you know, thyroid hormone is very important for normal health and development, particularly brain development in kids if it's low. And what we found in a cross-sectional study looking at the moms, where we measured the thyroid hormones and the PFASs in their blood at the same time, their plasma at the same time, was decreased free T4, which is one of the main thyroid hormones in the moms associated with prenatal PFAS. Well, we looked at the kids. It's routine to look at total thyroid hormone T4 in infants when they're born, and we found a association with decreased T4 in male infants. So you'll see this again, that there's seems to be differences between boys and girls that we don't really understand. Moving to some of the metabolic effects, we looked at obesity-related outcomes in children, and what we found is that prenatal PFAS, there were small increases in girls but not boys, and obesity-related incomes are things like body mass index, waist circumference, skin thickness, and something called DEXA, which is a really fancy way of measuring body fat. But the outcomes were not related to child PFAS levels. All right, so those are the more clear-cut things we found. In terms of metabolic function, we studied things like markers related to insulin resistance. So you see the word HOMA-IR, that's one of the ways you measure this. And we didn't find any evidence for adverse effects associated with PFAS. But amazingly, we found that children with higher PFAS had lower, meaning better, insulin resistance in girls, but not in boys. And again, this is a cross-sectional study, so you have to worry about things. In interpreting that a little bit, there is some toxicology data suggesting mechanisms that PFAS can work in opposite directions on metabolic function. So maybe that might explain what's going on, or again, because it's cross-sectional, we could have some reverse causation going on here. Now, lipid and cholesterol, this is one of the better studied effects in adults. There isn't very much information in children, so we were quite interested. And what we found was that prenatal PFAS was associated with improved blood lipid profiles in girls, but not boys. You can think of this as sort of the balance between good and bad cholesterol that you might have heard of. And with childhood PFAS, so again, cross-sectionally, we found an increase in both the good and bad cholesterol. All right, next to last outcome we'll talk about. Neurodevelopmental, so this is things where you can kind of think of it as like IQ tests and stuff like that. This paper was only published last week, and we did cognitive testing in early childhood, so about 3.2 years of age and on average, and mid-childhood, so 7.7. There was some evidence for decreased visual motor ability. This is things you can test by having kids like draw pictures and stuff like that. It might be associated with handwriting, so sort of coordination between eye and hand. Other results in the study weren't really very consistent, and sometimes they were associated with better cognitive outcomes, things like some memory tests. All right. I am involved with one more study right now. It's called the Diabetes Prevention Program Trial, which was a study of the prevention of type 2 diabetes among high-risk people. And we went back and we measured PFAS in the plasma of almost 1,000 people at baseline. They were followed up for 2 to 5 years, and this is what we found so far. At baseline, meaning at the start of the study, there were small associations between PFAS and markers of insulin resistance, but again, this is cross-sectional. We didn't find strong evidence of association between PFAS and diabetes incidence, although there was a little bit of evidence for the branched form of PFOA, but not the straight chain. Or we didn't find much evidence for prospective changes in glycemia indicators, that's things like blood sugar. So this is we've only been going on this study for a little while, and we've got some really interesting things coming out, so stay tuned on that. All right, so to conclude, Of the outcomes I've discussed today, if you're going to try to put this in the larger context, I think birth weight is the most established by the existing literature, and meaning this study and compared with other things looking at birth weight. If real, some of the things, differences we saw between effects in boys and girls are very interesting and kind of unexplained. We're digging into some more. There is a lot of good, high quality epidemiology underway right now, and I think we will know more reasonably soon. Like I said in the beginning, we're still sort of in the trenches on this. Now, again, we're mostly studying the older kinds of PFAS. Remember, it's a moving target. Finally, again, a reminder, environmental epidemiologists rarely, if ever, rely on single studies. Things can be inconsistent and don't always make sense in the beginning, and it typically takes a substantial body of work in order to sort of sort it out. And I didn't really try to present a full literature review today, just sort of highlights of where we're at on some of these things. And thank you very much to NIH, the National Institute of Health, and the Environmental Health Sciences branch for funding this kind of research. And there's references.
Okay, thank you, Tom, and thank you to our speakers. I see that there are a couple of questions that have been posted, some of which have been answered by our speakers. I think because there are people who take these calls by phone, they don't have the benefit of the Q&A text box. So we'll start with the questions that are being asked, and I'm going to start at the end and then direct it to our speakers who can respond. So there's a question to Dr. Webster. Do you know of any studies investigating health impacts of the entire cocktail of PFAS, or further information on that topic, as PFOA, PFAS aren't the only PFCs found when one or the other is detected? And this is a person who's living in Hoosick Falls. I'm going to leave it at that, if that's okay, to answer that part of the question, and then we can continue.
Yeah, so I'm actually quite involved with trying to understand effects of mixtures on health. I mean, to the degree that these things are all correlated with each other, we're kind of taking that into account. But of course, if we haven't measured it or can't measure it, it's difficult to know. So it's a really super good question. And the other thing you need to know is that there really has been almost no epidemiology done on the newer kinds of PFASs that we're starting to see in the water and elsewhere.
So along those lines, we just got a question again for Dr. Webster. Are you aware of epidemiological or toxicology studies focused on short chain PFAS compounds, C6 or C4?
I don't know of anybody doing epidemiology on C4. There are people trying to work on C6.
Okay, so we'll have to wait till those come out. I think this is for Richard. Someone asked, describe in more detail about the removal of arsenic from drinking water. I think that's in the context of PFAS, perhaps.
Sure. So again, it's certainly not my area of expertise. It was something we were concerned about and measured for to make sure that we were not providing that to our consumers of the water. There's two current, I think, beliefs on how the arsenic gets into the carbon, and it is in the carbon naturally. It's not a residual of anything St. Gobain did in Bennington. One is that it's an inorganic contaminant in the carbon and with a finite amount, and when you flush water through it, it then flushes through the system. I'm not in that camp, but that's certainly a plausible explanation. The other one is that it's based on water chemistry and whether the water is reduced or oxidized. Many metals are mobile when they're in a reduced condition and not when they're in an oxidized condition. And so after you flush enough water through the system, that water is oxidized, the carbon then becomes oxidized, and the arsenic becomes immobile and sits there not harming anyone. I hope that answers that question.
Okay. Another questions are coming in. So let's see. can you put the methods for detecting PFOA, PFAS in NHANES data into context? Do they look at total inorganic, organic fluorine metabolites of PFOA, PFAS, or are they detecting PFOA, PFAS compounds?
This is Tom. I mean, they're looking at specific compounds. They're not looking at total organic fluorine.
Okay, I'll keep going with Tom and then back. Please speak more about joint toxicity. Are there bad PFAS actors that might be interacting for key epi endpoints? And please comment on whether multi-pollutant PFAS models should be done in all studies.
Yeah, well, the problem is a lot of these things are correlated with each other, so it's difficult to at least in epidemiology, it's difficult to sort them out. I think we're working on new statistical methods to do that, and we're going to need toxicology work to help really figure that out.
I'm jumping to the beginning now. Regulated entities often question the need to report results for all PFAS because most regulatory agencies only have standards or guidelines for 2 or 3 of them. This will be to Nancy.
Yeah, this is not an unusual problem. However, as we've been hearing from Tom, and the research area is growing, and the toxicity and the epidemiology of a wide range of compounds. So if you're only looking at a small subset of the compounds, when new information becomes available, you would have to go and resample and reanalyze the samples at additional cost and expense and time to get more information. So why not get as much information as possible, as easily as possible?
Another question, you mentioned that the spent GAC is incinerated. How effective is that in destroying PFAS compounds? I guess that's Richard.
Sure. Well, it's certainly something we were concerned about, not just moving our problem to some other location to be deposited. So, we reached out to Calgon, and they assured us that it's being incinerated at a very, very high temperature, high enough to break the bonds between the carbon and the fluorine and destroy the PFAS compound. So, we feel pretty good based on that, but I certainly don't have any firsthand information other than other regulators who have also looked into it, have reported the same thing.
With the groundwater health advisory levels so low, they're down in the 70 parts per trillion and lower. At what levels are the soil cleanup levels coming in? Are you aware if soil cleanup levels are being implemented for any project? Richard?
It's probably for me again, yeah. So there are a couple of issues relating to soil contaminant levels. One is the direct contact level, which we studied or looked at very early on in Bennington. Our health department set a direct contact standard of 300 parts per billion. I just want to make sure that's clear. That's parts per billion, not parts per trillion, which is what we're talking about for drinking water, for direct contact. And I think that's mostly based on the amount of soil that you actually ingest compared to the amount of water that you ingest daily, although perhaps Tom could tell us more about that than I know. But anyway, based on that standard, we had no soils that were anywhere near the level of concern in Bennington for that direct contact. Now, there's a secondary issue with soil, which is what's the risk of groundwater contamination from the soil? How much of what's in the soil leaches from the soil and into the groundwater, which then could or is drinking water? We were less concerned about that in Bennington because we already knew we had a significant groundwater contaminant issue. However, what happens if that soil gets removed and taken to another location? What's the level that you would need to look at for causing a problem at that new location? And many states are looking at that issue, and standards are beginning to come out on. In Vermont, we don't have one, what we did in Bennington, because we are removing a lot of soil and needing to relocate that as part of the waterline extension work, is we're keeping all that soil local, right, where we already have a problem, and where we're less concerned, not that we're not concerned about the groundwater, obviously We're very concerned about that, but we're less concerned because it's already contaminated, and that we are going to be bringing either have or are bringing municipal water to all the residences there. And that will make the movement of that soil from one location to another inside that small area not that much of an issue. So there are standards that are being set, and they're much lower than the direct contact standard.
Okay, thank you. There's so many more questions, and in the interest of time, let's take one more, and then we can respond to those that are posted. We can respond by text. Is that, I think that works. So for drinking water, should we be concurrently testing for total PFAS? Richard, what are you doing in Vermont? And I think this goes back to what Nancy had said about getting information as
Sure.
As more states.
So again,
yeah.
I mean, Tom may know more about this than I know. It's certainly not my area of expertise, but what I can say is, and it goes back to the regulatory question, in Vermont, we report all of the analytes that are tested for. We actually send a copy of the lab results with it, and put a cover sheet on it that explains what the numbers mean based on the regulatory number versus what the other numbers are, even though we don't know what that means regulatorily or health-wise. So people have that information if they need it or want to use it at a later date. As far as sampling for all analytes, well, obviously you could only sample for the 14 based on the EPA method. If you wanted to do like a top assay to look at all fluorine, I don't know what information that necessarily would provide you. It could be done, but again, since we don't have standards, I'm not sure what that information tells you.
Yeah, and I'll just add, this is Nancy, is that these other methods really are look at not compound specific. So it may tell you that just doing the 14 compounds, you've missed some fluorine content in your sample, but you wouldn't know what that fluorine was attached to. So really, for drinking water, you must do method 537, which only has 14 analytes. And unless you get a dispensation to increase that list, that's the only analytes you can report at the time.
So I think in the interest of time, we will take the questions that have been sent in and respond offline to those questions. And I want to thank the speakers. Clearly, we could go on for quite some time, but we only have you for an hour. So thank you very, very much. I'll turn it back to Maria.
Great. Thank you so much, Wendy. A video recording of this webinar will be available on the CHE website soon, and tomorrow you will receive an email containing a link to the recording, as well as to the survey that Wendy mentioned. CHE's next call from the CHE Alaska Partnership will take place tomorrow, May 2nd, and is titled, Nursing the Earth Back to Health, Personal and Policy Strategies for Preventing Breast Cancer. You can RSVP through CHE's website, healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these CHE Partnership webinars, bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers for taking the time to present today, and Wendy for her excellent moderation. Thank you so much for joining us, and have a great day.